there's only one handout, and this basically comes from your textbook. There are a couple of pages that I wanted to have and pass out so that it's kind of like a cheat sheet, or it's a list of characters. Maybe another way to describe this is we've got a list of characters here that we are going to introduce. And on one side, you've got the phylogenetic tree that shows their evolutionary relationships. And you'll notice they're kind of broken up into different clades. Different colors you can see are given different clade names. And we're going to look more in depth at each of those clades. Clades, remember, are just a common ancestor and all of their descendants. And we can see smaller clades nested inside larger clades. And what you're holding here is as a cladogram of the entire eukaryotic branch of organisms. Your list here is a little bit larger than the one that we're going to see in the slide today. I'm just going to pull this up real quick. You have on yours, let's see what we have here. This is a slightly older version. So one difference being that this group of Chrome Alveolata has now been separated into two separate clades. It's the same evolutionary relationships. They were already distinguished in a previous version of a textbook, but if you look at your version of the textbook now, they've totally separated this Chrome Alveolata group into the Alveolates and the Stramina Piles. So you have now in separate colors Alveolates and Stramina Piles. You can see that if you zoom out a little bit, that both of those are kind of part of the same larger clade. So we'll see this when we get into the slides, that the evolutionary relationships between protists is an area of intense research now because we've got the molecular tools to do this. Before, they had to study organisms' life cycles to see who was able to reproduce with who. And with a bunch of these microorganisms, a lot of that wasn't understood. So having molecular tools to be able to get in there and find these relationships has been really helpful. It's why within the last 10 years, when they come out with a new version of the textbook, it's typically this chapter on protists that has been revised. So we're going to go through and see examples of each of the 7 groups that we have to know. If you'll just do me one favor, if you have that list that you have in front of you, that's all of the eukaryotes. And I'm jumping around to some slides here. I'll jump to this slide. I know there's a lot of pictures on it, but this is basically the clade of eukaryotes. It includes the common ancestor of eukaryotes and all of their descendants. So this is a true clade, but what I want you to do, what I was about to ask you to do here, is to maybe temporarily, yeah, you'll need one of those. If you can just temporarily maybe put a little line through or a little mark or something through the groups on that page that include animals, plants, and fungus. Because protists, we're going to learn, are not a true clade. They're going to include a common ancestor and some, but not all of the descendants. We're going to exclude plants, animals, and fungus. So the red lines here are indicating that we are excluding them from consideration as protists. What they have in common with protists is that fungus, animals, and plants are also made of eukaryotic cells. So the rest of these things are all the organisms comprised of eukaryotic cells, not a plant, an animal, or a fungus. So if you cross off animals, plants, and fungus from your list, the remaining 7 clades are the 7 clades of protists that we need to be aware of. I can already see some exam questions showing up where these 7 clades are the options, and I'll be describing organisms, examples of an organism, from one of those 7 clades. Just in time, there's your cheat sheet. You'll notice on one side you've got the evolutionary relationships of all eukaryotic organisms. Today we're talking about protists, so this is kind of the same thing we're looking at, this clade of all eukaryotic organisms. And what I was just asking everybody, if I have a pen, I'll give you one. Oh, here you go. It's a red one, but it still works. I was just asking everybody to maybe draw a line through the box that includes fungus, animals, and plants, because those are not protists. And I'm just wanting to give you this chart that shows evolutionary relationships between all the protists. So again, all we have, I might have just launched into this, but all we have today is our lecture on protist. And I guess I should kind of set this up by saying Protists are extremely diverse, and that makes this lecture particularly difficult because we talk about such a wide variety of things. You know, if we're talking about just plant vascular tissue later on, it's going to be easy to focus. We're just thinking about plant vascular tissue. But now we're thinking about organisms that are fungal-like, some are animal-like, some are plant-like. It's really broad, so to kind of help out with that, that's why I wanted to give you this chart. So now we've kind of crossed off animals, plants, and fungus. We've got 7 groups that we're going to look at, and I'll give you an example of each of those 7 groups. We'll try and include pictures of all of these. But that's all we're doing for today. The quiz that is up on D2L, it was up last week. It included prokaryotes and a couple of questions about viruses, viroids, and prions. And then the new stuff, which we talk about this week, the questions have already been up there, are about the protists. So we should separate prokaryotes and protists from one another. And on Wednesday, we will do our lab, Protist Diversity lab activity. We're going to be working with microscopes. We're going to see examples of most of, in fact, all of the protists that we have on the screen, we're going to see examples of. So today we really kind of set that stuff up, go through our cast of characters, and then Wednesday we're going to kind of get our hands on those. And I didn't write it here, but there's going to be a post-lab quiz following Wednesday, whatever we see there. So is everybody doing okay? That's our introduction to Protist. All right, I'm going to go back to the beginning. It's only like 40 slides, so it shouldn't take too long. And I do have the exams to give back. Everybody's taking their exam now, so I'll be able to give that back to you. I might not have graded yours if you took it today, but I'll have it for you on Wednesday. Hopefully that's all right. So today we talk about protist evolution. Wednesday we talk about protist diversity. We'll start with this picture because it's what we saw last time, and our lecture last time was all about archaea and bacteria. Some of the first quiz questions were about differences between archaea and bacteria. We listed some similarities between the two. But this time we are talking all about the eukaryotes. And since we're going to introduce protist, like I just pointed out, you have this eukaryotic cladogram in front of you. And if you draw the line through fungus, animals, and plants, what is left over are the protists. Protists, even though that word sounds like prokaryotes, and I know I've messed that up already a couple of times, prokaryotes are these tiny cells that don't have any organelles, and we said that prokaryotes are about the same size as an organelle inside of a eukaryotic cell. Protists, on the other hand, like this Euglena, protists are made out of these eukaryotic cells. So they're going to have organelles, they're going to have internal compartments, they're going to have a nuclei. They're larger cells. So even though those names sound very similar, one is made out of eukaryotic cells, the other one prokaryotic cells. If we remember from 1406, I feel like we talked about this in the Unit 2 of 1406, at some point when we introduced prokaryotic and eukaryotic cells, we saw the formation of the first eukaryotic cells, a cell that has a true nucleus. And that nuclear envelope really just comes from infolds of the cell's plasma membrane. You can see this plasma membrane start to have these internal folds that ends up sectioning off all the genetic material of this cell. Now it's no longer floating around in an open swimming pool. That genetic material is encapsulated in its own little membrane. Now all of the cytoplasm separated from the genetic material, the cytoplasm is free for other chemical reactions to take place. And sure enough, that's what we start to see. These, what we consider the host eukaryotic cells, a cell with the first nucleus, start to engulf other prokaryotic cells as a fuel source. Here's just that picture that shows one of the first endosymbiotic events. This is the only handout that we have today. One side shows the phylogenetic tree of eukaryotic organisms. We're talking about protists today, which is going to be everything except the animals, the fungus, and the plants. So I was asking people just to maybe cross a line through plants, animals, and fungus. And the remaining 7 categories on there are going to be the 7 clades of protist that we should get introduced to today. Most of these protists, since they're made out of eukaryotic cells, when we look inside of them, we're going to see internal compartments. On this model, I'm not exactly sure. Maybe those orange dots are meant to be the mitochondria, but this just shows one of the first endosymbiotic events in which a prokaryotic cell was engulfed by a eukaryotic cell and not completely destroyed, but instead put to work. This relationship got started between this prokaryotic cell, whose job it was just to break down glucose and make ATP. It's what it did all by itself. Now it's engulfed by this eukaryotic cell, and the eukaryotic cell uses it to break down its glucose. So we said we come from a lineage of cells that arose after this first endosymbiotic event, because all of our cells have mitochondria. But there was a second endosymbiotic event. There's the little mitochondria close-up picture of it. It has its own fluid, it has its own ribosomes, it has its own DNA. Not that we can really see the DNA in there, but the DNA would be in little tiny circular loops like it would be in a prokaryotic cell. I said there was another endosymbiotic event in which another prokaryotic cell got engulfed, and this particular prokaryotic cell was photosynthetic. And this is what led to the lineage of plants. We're going to see that some cells, even this Euglena, arose after this second endosymbiotic event. So these cells have both mitochondria and chloroplasts in them. This is what we would consider autotrophic. They can not only make their own sugars, but they can break them down. Our cells having just mitochondria, we're heterotrophic. We can't make our own sugars. We need to ingest that from somewhere else. Whether we're thinking about the chloroplast or the mitochondria, I said they both kind of have they still retain their primitive characteristics from when they were their own separate prokaryotic cell. Here's a close-up picture of one of the chloroplasts. It has those stacks of thylakoid discs. When we look inside a photosynthetic prokaryote, we see that they both have the same DNA that's in a circular loop. If we were to draw these things out on a phylogenetic tree, you would see they are sister taxon to one another. So we can trace lineages of prokaryotic cells and organelles inside of cells. And this particular organelle shares a recent common ancestor with photosynthetic prokaryotes. And I talked about that last time. I think I might have gotten a little bit on a side note in our last lecture when we were introducing the types of the various ways in which prokaryotic cells could get energy from the environment. We talked about photoautotrophic prokaryotes that are just like plants. We talked about they get their energy from the sunlight and their carbon from carbon dioxide. We talked about photoheterotrophs, and we talked about chemoautotrophs and chemoheterotrophs. And somewhere in there, when we were talking about the photoautotrophic prokaryotic cells, I was all excited because those were the cyanobacteria, the first prokaryotic photosynthetic cells. And what's kind of interesting is that, well, just to jump to this, what we see is that photosynthesis has not been reinvented in the other cells, the plants, the other protists that are also capable of photosynthesis. Instead, that first photosynthetic prokaryotic cell has just been engulfed and has become an organelle of these lineage of cells. So it's the same process that's only really been figured out once, that's been really hijacked by these other cells. And I'm going to go back just to the slides that I skipped over. Only been invented once, like I said. We mentioned at least 5 lines of evidence for that back in 1406. The fact that those prokaryotic cells and organelles inside of a eukaryotic cell are about the same size. We said those prokaryotic cells and these cells, organelles, have their own ribosomes. They have their own DNA. That DNA is still active. It still makes proteins. So again, prokaryotic cells and organelles like mitochondria and chloroplast are still active. Were once their own prokaryotic cells. I was just talking about this primary endosymbiosis where there was a host, kind of a primitive eukaryotic cell that has a nucleus but doesn't yet have a bunch of internal organelles. This used to feed on other prokaryotic cells, and one of those prokaryotic cells that got engulfed found a home and got put to work. So this is what we were just talking about, this primary endosymbiosis where mitochondria and chloroplast were first engulfed by eukaryotic cells. If we can zoom into this, we're going to see that that process of endosymbiosis, a prokaryotic cell getting engulfed and put to work, has happened a second time and on a larger scale. These are now eukaryotic cells, and just to zoom into this a little bit, this is just a normal prokaryotic cell that had figured out photosynthesis. It's the same type of cyanobacteria that we see in lab. Some of those prokaryotic cells are using different wavelengths of light, and I have a little snapshot over here of the first step in photosynthesis. Last semester we had to remember the light reaction and then the light independent reaction where carbon dioxide got put together into a glucose. I just wanted to remind us that the first step in photosynthesis, where sunlight, it was just that photon of sunlight that was used to split a water molecule. And then half of that water molecule, this also was helpful because we had a random lecture last time where I was talking about cardiac physiology, and you saw it was difficult for charged particles like calcium or sodium or potassium to cross the plasma membrane just anywhere on its own. There had to be a certain channel, a protein channel, that opened up that let those charged particles across. We remember that in photosynthesis, that sunlight split water molecules, and then the hydrogen ions, those positively charged hydrogen ions, couldn't just leave that thylakoid disc anywhere. There had to be a special gate that opened up that let them flow from high to low concentration. And as they were making their way out of that thylakoid disc, they spun that little turbine and generated ATP. Where I'm going with this is the same process that's going on in the cyanobacteria. It's just that some of those photosynthetic cells are capturing different wavelengths of light to split that water molecule. So some of the photosynthetic cyanobacteria that were capturing longer wavelengths of light end up leading to this lineage of red algae, and we'll see wavelengths, shorter wavelengths of light are picked up by the green algae. So we'll see red algae when we talk about it today. It's typically in marine environments that are in a little bit deeper depths. Those are longer wavelengths that can penetrate deeper down into the water column. So anyway, you've got red algae and you've got green algae. Again, these are eukaryotic cells. They've got a nucleus. They've got an organelle inside of them that's capable of photosynthesis. But we can see red algae and green algae also get engulfed by another eukaryotic cell. This is what we call secondary endosymbiosis. And whether it's green algae that get engulfed and put to work, or red algae that get engulfed and put to work, this leads to some of all of the diversity that we see in protists. So again, these are eukaryotic cells. They've got their nucleus. They've got organelles inside of them. Some of them are plant-like because they are autotrophic, but they're also animal-like in the sense that they can move around. We should see examples on your list. If you flip on one side, you can see the 7 clades that we have to know. But on the back side, it's not in color. So look at your textbook if you want the color version of this table. But it gives you kind of a quick reference in terms of the names of the organisms within this group, whether they are heterotrophic, autotrophic, what kind of diseases they might cause. This is where I'm going to get the questions, or the ammunition to build the questions for the quiz and for the exam. So we're about to take a tour through some of these different types of protists, and we're going to look at pictures of these as we go through. I mentioned the lab that we do on Wednesday. We're going to see examples of each one of these organisms that made it onto this slide. I think we even have more than made it onto this slide. So let's start at the beginning. Before we look individually at the different protists, I'm going to mention some general things about protists. And I've said some of this, so I can go through kind of quickly. The first thing that we want to think about when you hear the word protists, because it sounds an awful lot like prokaryotes. These are not prokaryotic cells. We said they're all made out of eukaryotic cells. Anything that is comprised of eukaryotic cells that excludes, that is not a plant, an animal, or a fungus, is part of this group that we call protists. So if you go back to that big chart, like I said, and you cross off 3 of those boxes, we cross off the plants, we cross off the fungus, we cross off the animals. What we have really created is what we mentioned last time in Unit 1 as a paraphyletic group. A true clade is a monophyletic group. It would include the common ancestor and all of these descendants. But when we leave out some of those descendants, we've created a paraphyletic group. It's going to include some, but not all of the common ancestors. Just saying that protists are not a true clade, they're a paraphyletic group. And since we have all these new molecular tools, it's really this chapter in the textbook that seems to get revised most frequently. This is a picture of one of these protists that we're going to see. It's a single-celled organism, and it's basically a tiny little amoeba that is defenseless. And so what it builds to kind of protect itself is this little glass shell. It's called a test, not the type of test that you just took recently, but it's basically a home. And this is a test or a shell that's made out of silica, so it's a glass structure. And it has to reach out between these little openings, these little pores in the glass structure for food particles that are in the water column. Those little arms, you can see one reaching out there, are called pseudopodia. We'll see that one in a second. Some other general characteristics about protists. They're typically single-celled, but we're going to look at a couple of examples of what we're going to call colonial or multicellular. For the most part, all of these protists are small enough that there is no internal system set up. Like, we are large enough now that all of our cells cannot get enough oxygen and get their carbon dioxide washed away in time. So we have things like a circulatory system and a respiratory system set up to get gas like oxygen into the bloodstream, into the cells, and wash carbon dioxide away. These cells, since they are tiny enough, there's plenty of surface area for gas exchange to diffuse in and for the other gases like carbon dioxide to diffuse out. So we don't see any complex internal organ systems set up. Most of the gas and the waste exchange can take place just across the surface of the plasma membrane. We will see some of these things have some internal organelles set up that are going to allow for water balance. Some of those circles that you see right there are going to be what we will call contractile vacuoles. We'll see these on Wednesday. These cells, remember animal cells, it's problematic. It's more problematic for an animal cell to wander into a hypotonic solution. We mentioned this last time because in a hypotonic solution, an animal cell is going to start to take on water, and there's no cell wall there that's going to prevent it from swelling too much. So animal cells that start to take on too much water have a real threat of rupturing and exploding. So to prevent too much water being taken on, we're going to see these cells have organelles that are going to push out excess water. So they're capable of water balance. Again, in addition to some of the water that's pushed out, we're going to see ions are pushed out as well. We had mentioned in our cardiac physiology lecture that we randomly had last time that there are voltage gates that are going to let, on the plasma membrane, there are protein channels that let ions cross, specific ions. So it's nice that we got to at least have that lecture last time so that you get an example of a specific example of the types of proteins that you see on a plasma membrane, rather than just saying that there are proteins that regulate ion balance on either side of the membrane. So again, I will probably refer back to that cardiac physiology lecture when we see things like this. It's still present even on single-celled organisms. We're going to see proteins along the plasma membrane determining what ions move in and what ions move out. Okay, so here's another distinction. We were seeing that most of these protists are going to be, the entire organism is just made out of a single cell. Most of them are going to be unicellular. But there will be a couple of examples that we're going to see in our activity on Wednesday of colonial protists, and we're also going to see multicellular protists. Those two terms, the distinction between them is not immediately obvious. This is an example of a colonial protist. And you might be thinking, well, there's more than one cell there, and you would be right. But the reason it falls under colonial rather than multicellular is that anything that's comprised of more than one cell is no longer unicellular. But what makes this colonial is that each cell is doing the same thing. Notice how each one of those cells are identical to the other cell. There is no cell differentiation or cell specialization. You're multicellular. You've got cells that specialize in lining the surface of the body or lining passageways, cells that are specialized for sending electrical signals or contracting or connecting things. You've got cell specialization. So anything that's multicellular, we're going to see cells differentiating from one another. Anything that's made out of a bunch of identical cells is just going to be colonial. So this is a colonial organism. In terms of nutrition, we're going to see a mixed bag in terms of protist as well, because some of them are still from that lineage of cells that only picked up mitochondria. They don't yet have chloroplasts, so they're heterotrophic like us. Others have picked up chloroplasts, so like plants, they are autotrophic. And just to make another distinction, of the ones that cannot make their own food, of the ones that are heterotrophic, we can distinguish two different types of heterotrophs. We belong to the first type of heterotroph, these ingestive heterotrophs. All of our digestive enzymes are inside of our body. So if we are hungry and we want to not be hungry later, we're going to have to get something inside our body so those digestive enzymes can break it down. Fungus are going to be an example of an absorptive heterotroph. We're going to see some protists like this as well. They have their digestive enzymes that actually leave the cell, and all of the digestion happens outside of the body, outside of the cells. And then the cells basically absorb the resulting nutrient-rich goo that is left over after those enzymes. This is what fungus do. They secrete these digestive enzymes on dead wood, and then the wood kind of gets dissolved and broken down, and then the cells absorb that nutrients. Absorptive heterotrophs. Sometimes the digestive enzymes are on the outside. And then we'll see things like Euglena, this thing that I keep holding up, that's a little bit of both. It's part animal. It's capable of moving around and breaking down its own sugars, and it's got chloroplast as well, so it's capable, it's plant-like in the sense that it can make its own food. The terms, these are old terms, so we won't really use these, but you still hear them, so I just thought I would throw this out there. There's a research methods class that we're doing, and some person in that class has a horse that is suffering from this disease that affects the neural tissue of a horse. And this student wanted to find out what is it that's exactly causing that problem. Is this a bacteria? Is it a virus? Is it some type of protozoan? And they found out that it was a protozoan. It was something that they were getting on the diet. It was a protozoan that was affecting the neurological tissue of this horse. Protozoan, that is an old term that means animal-like protist. So these are typically those heterotrophic protists. They can't make their own food, so they're going to go get it. They're going to steal it from something else. So protozoans, when you hear that word protozoan, it's an animal-like protist. There are plant-like protists that you typically get one blanket term that describes all of the plant-like protists, and they're just called algae. But we can see there's a bunch of different types of algae: green algae, red algae, brown algae, golden algae. All of these, when you hear the word algae, you're thinking a protist that is like a plant, it's photosynthetic. But just to make a distinction, since we are now getting into eukaryotic cells that are photosynthetic, that are not plants, we need to make a distinction between eukaryotic cells that are capable of photosynthesis, the algae, the plant-like protist, and actual plants. So I know we will get into an entire unit on this, but anything that is an actual plant, we're going to see has adaptations to life on land. Anything that is considered an algae, whether it's a green algae, a red algae, any of those types of algae, these are aquatic. They have no adaptations to life on land. I'll extrapolate on that further as we get into there. And then just another thing that we'll see, we have this one in lab. It's a slime mold. It looks like a fungus, it acts like a fungus, but it's actually a protist. This whole thing, and it was on a petri dish, I think I might have brought it out last time. If not, I'll go grab it. it's the size of an entire petri dish, and the whole thing is just one cell. And if you look at it under the microscope, you can see that through the little channels there, it has cytoplasm that's just flowing like a river. And that cytoplasm flows in the direction of wherever there are nutrients. If cytoplasm is flowing in one direction and there's really not any nutrients over there, then that's going to dry up, and these veins that lead towards nutrients are going to continue to grow. So this thing kind of moves through the forest floor and breaks down dead plant tissue. The one that we have in lab is actually moving around agar, this gel-like substance, and it's eating oatmeal. Anyway, we'll see on Wednesday a whole diversity of protists, some that are plant-like, some that are animal-like, and some that are fungal-like. But you have this chart in front of you. This is a slightly older chart, like I'd mentioned. This one of the differences is that it has the alveolates and Stramina Piles still part of this one big superclade. Your newer chart now has those separated into two different colors. You can still see that these are there. Right here, this word alveolate, you can see that line is trying to represent a common ancestor. And if we include ciliates, AP complexions, and dinoflagellates, that would be this common ancestor and all of its descendants. So alveolates would be its own clade. Stramina Piles is its own individual clade, kind of nested inside this much larger clade. So I put this, it's the same slide. I just wanted to add some numbers to it so that we know what qualifies as a protist and what does not qualify as a protist. So I asked you to draw a line through the box that included plants. We're going to see plants have a recent common ancestor with green algae. They share a more recent common ancestor with green algae than they do with red algae. And you might be wondering what the difference between chlorophytes and chyrophytes are. They're both green algae. And one of those, these chyrophytes, I apologize for that thing wiggling. I think it's the wind. Chyrophytes have at least one adaptation to life on land. All land plants are going to have special adaptations to life on land. So again, green algae, red algae, any of these things that we consider just algae have no adaptations to life on land. So we've crossed off plants. We've also crossed off the animals and the fungus from our list. But as we go forward, we're going to see examples of these excavates. We'll see alveolates, Stramina Piles, rhizarians. There are two examples of rhizarians that we're going to see. We have examples of red algae and green algae, and a couple of amoebas. So here we are with some actual pictures. I didn't miss it. Everybody has a list, right? I gave you one of those, right? Okay. We're starting at the top of that list with the excavates. You can see you've got 4 examples of excavates in there, and the first one on your list is the Diplomans. One particular example of a Diplomans is Giardii. This is a common parasite that you find in fresh water in streams. If you go camping and you fail to boil the water properly, it's typically this organism that gives you intestinal cramps and diarrhea and a lot of pain. It affects the intestinal tract of mammals. What I also think that's curious is that when we're looking at these things on slides, we're seeing these organisms really in 2 dimensions. So you have to almost kind of imagine this 3-dimensional picture. This is an electromicrograph, kind of a scan of this organism, and all of these excavates, they kind of have this little groove like something has been excavated out of the center of them. This one also has these little flagella that it can use to move around. It doesn't have chloroplast, it's not photosynthetic, so it's going to have to get its nutrients from something else, and this gets its nutrients from breaking down the lining of mammals' intestines. So boil your water, or Giardii is a likely candidate of giving you some discomfort. So that's one excavate. Our second excavate is a Parabasalid. This is the second one on the list. Parabasalids are also heterotrophic. You can see they don't have any chloroplast. They're not going to be able to make their own food, so they're going to have to break something else down. And again, also, we're seeing this thing. This would look like a completely furry I don't know, a furry little spherical or oblong structure. We're seeing, again, just a two-dimensional sliver of this organism, but you can kind of see how fuzzy it is. These are found in the digestive tracts of termites. There used to be a lab that we would do where we would pull these termites out and they would all be wiggling around in a petri dish, and you would have to use your scissors to just pull the head off of the termite. So it was kind of cruel that you would kill these termites, but then like a tube of toothpaste, you would squeeze all of the goo out of the termite. And it's all the stuff that was in its digestive tract. And under the microscope, if you would look at the stuff that was in the termite's belly, what you would find are all of these tiny protists. Even a termite is like a community of organisms. It has in its digestive tract these protists, these animal-like protists, that are capable of digesting the wood, the cellulose that the termite eats. It's crazy to think, just like we talked about the cow last time, it goes around eating plant material, but it doesn't have any enzymes to break down plants. It relies on the microorganisms in its digestive tract to break down those plants. The termite is doing the same thing. It eats a bunch of wood, but it doesn't have any enzymes to break down wood. The termite relies on these protists that are capable of breaking down the wood and converting it into some type of nutrient-rich sugar that then the termite can absorb and gets nutrient from. So we find these things in the guts of termites. We said they break down cellulose. Trichonympha is the genus name. It's our example of a Parabasalid. Third thing on the list in terms of excavates is the Euglena. And these, out of any of the excavates, these are the ones that we have live examples of in lab because they are the autotrophic ones. These are capable of making their own food, and we can see that because of the green color. They're packed full of those little organelles that are doing photosynthesis. They just have to have exposure to sunlight. So we can see that other organelle in there that we said was the eye spot. It's this eye-looking structure on the model. We said it equates to about 10% of your eyeball, so it can't form an image at all, but it can sense the presence or absence of light. It can move towards bright light or away from bright light and dial in how much photosynthesis is occurring. So part plant and part animal. It has that little flagella that it can move around and adjust how close it is to the light. These are all over pond water. In fact, one of the things we're doing on Wednesday is I have in lab on the cart, we've got fresh examples of a bunch of these different types of protists. So we're going to make our own slides. We're going to see what these things look like from the labeled container, and then we're going to sample some random pond water that I'm going to bring in. And we're basically going to create a list of, we're going to take pictures of a certain group of protists that, it comes from a jar. We know what we're looking at, but then we're going to get some random pond water and we're just going to see what we can find in there. And hopefully we see some things that look familiar to us. Anyway, Euglena, I bet we find some of this stuff in the pond water. It's in almost all aquatic food chains. The last example of excavates that we see, our first group of protists that we are introducing, is once again one of these organisms that is not capable of making its own food. It is heterotrophic. It has to go break something else down, ingest something else. And a couple of these examples of, we will see the Chinoplastids, the last group of excavates that we have on our list. These are typically parasitic and can cause problems for people. The first picture that we have up here is, those are tiny red blood cells, and between those red blood cells, those are the protozoans, the animal-like protists that are attacking those red blood cells. Here's the same picture. It's just an electromicrograph scan that has a little better color representation on it. We can see red blood cells. We can see those trypanosomes. Those are the protozoans. It's a eukaryotic cell. It's a single-celled organism, and it's attacking those red blood cells. So trypanosoma are these squiggly little structures. We've got a slide in lab that we will see. They are one of the things that causes African sleeping sickness, and they're brought by this little fly. It's a biting fly, and it delivers this trypanosomes to people. There are other trypanosomes that are parasitic. The pictures that you see down here, this is a flesh-eating disease called leishmaniasis. And it's not bacteria, it's not virus, it's caused by a protozoan, an animal-like protist, a type of trypanosome. So excavates, just on our blue list, we can see we've got 4 excavates to be aware of, and only one of those 4 are we going to see in lab, because that was the only one that was autotrophic, that was plant-like. The rest are animal-like, they are heterotrophic, and typically they feed on some part of us. So the next group that we're going to look at is the purple group that is on your list. We're kind of not going in an exact order, but if we go down on your list to the purple group, that is the Stramina Piles. And there are 2 examples within the Stramina Piles that we're going to see. Actually, in lab on Wednesday, we're going to see a water mold, we'll see a brown algae, and we'll see diatoms. We'll see slides of each of those. But in our lecture, I brought examples of diatoms. The first one, these are all marine, aquatic, photosynthetic structures. This is a single cell. It's capable of photosynthesis, so these are all autotrophic. Like I said, they're single-celled organisms, and they are very delicate. So to help protect themselves, they encase their single cell in what's just silica material that they grab out of the marine, I don't know what word I'm looking for here, marine environments. If we're in an aquatic marine environment, in the water column itself, there are minerals that are dissolved like silica, also calcium carbonate. Some of these protists are going to grab that calcium carbonate and make their little protective home. These organisms are grabbing silica from the water column and building their homes. So their little shell or their little house is made out of silica. We have some slides on Wednesday that show the diversity of different types of marine diatoms. There's a bunch of different shapes that they can build their glass house out of, and we'll see a variety of those. So diatoms are one example of these Stramina Piles. The other example that you can see there is brown algae. So brown algae is, maybe what's noteworthy about brown algae is it so far is our first example of a multicellular protist. Multicellular means that we're going to see cells that have differentiated for different purposes. And look, this organism has some cells that have differentiated into what look like a stem, and other cells that have differentiated into what look like a leaf, and even other cells that have differentiated into what look like little floating pods. And in fact, this is, once again, we're seeing cell differentiation, cell specialization, so this is a good example of a multicellular protist. Brown algae or seaweed, this is common even on the Texas coasts. If you've been there, it seems, I don't really get to go very often, but it seems like recently when I've gone down there, there's been a bunch of seaweed that has come onto the beach. And that's not all bad, because I like to pick through that. That's a whole ecosystem. You find these sea slugs and all kind of things that are in there. But if you go down there and you grab some of that seaweed and you start flipping through it, you'll see that there are cells differentiated for becoming what we call these leaf-like structures. It's not a plant. It's an algae, so we can't use the plant terms to describe these differentiated parts. So even though they do the same thing, this blade functions the same way as a leaf functions in a plant. They're broad structures that catch a bunch of sunlight that try and maximize photosynthesis. This structure here called a stipe, it behaves the same way a stem would behave in a plant. It's not a plant, so we can't use the word stem. It gets its own term, stipe. And then these structures down here on brown algae called a holdfast just anchor this to the substrate. Looks like it might be on some type of coral reef currently, but whatever it's anchored to, these holdfast are representative of the roots. Again, it's not a plant, so we can't use the term roots. We would use the term holdfast. But you can see cell differentiation for anchor points, for supporting the photosynthetic pods. Even on this previous picture, we saw some cells specialized into floats. that carry those blade parts up close to the surface. Again, maximizing photosynthesis. So brown algae, our first multicellular protist. I said there was only 2 examples of the Stramina Piles, diatoms and brown algae. So the next of our 7 clades on the list, we'll move to our 3rd clade that we're introducing, and that's the Alveolates. Again, it's kind of right next to Stramina Piles. These used to be grouped together. Alveolates, we're going to see the first example of alveolates are the Dinoflagellates. So I'm kind of going in random order once we get inside the alveolates. Dinoflagellates are going to be the only ones of the alveolates that are plant-like, that are autotrophic, that are capable of making their own foods. They're green, so maybe that's the first giveaway that they are capable of photosynthesis. They have this groove that kind of goes all the way around their body, and there's a little flagella that is found within that groove, and that helps them move around in the water column. So think marine environments. These are single-celled organisms, and they're capable of photosynthesis. And maybe the story behind these dinoflagellates that you see in Texas news articles, typically you usually see this in September and October, and what's going on is usually in September and October we get a rainy period that comes through here, and in the Rio Grande Valley there's a lot of agricultural farms, and so during our rainy seasons, this happens on golf courses too. They'll put a bunch of fertilizer down, and then the next weekend there'll be a big rainstorm, and all that fertilizer gets washed into the surrounding ponds, and then you have this explosion of green algae that you see in all of the water. And that happens, like I said, in the Gulf of Mexico as well. Following rainy seasons, a lot of that superficial fertilizer gets washed off into the Gulf of Mexico, and it supports the explosion of dinoflagellates. These things, again, just like plants, they feed on that fertilizer, and when their numbers explode, this is problematic because what happens, they are photosynthetic, so they start to produce a bunch of oxygen, and the water temporarily has a bunch of oxygen in it, and so you'll see these fish that move into that area. They're loving it, they're loving this oxygen-rich environment, but the fertilizer is what supported this explosion, this boom of dinoflagellates. And if the fertilizer doesn't keep coming, which it never does after those big rainfalls, this entire population of dinoflagellates goes through a boom. And as the dead body, they're no longer producing a bunch of oxygen, they're dead, you get the bacteria, the microorganisms that break down these dinoflagellates. And that the breakdown, the dead bodies, the decomposition of these dinoflagellates, that is an aerobic process that uses oxygen to break these things down. So the bacteria, the microorganisms that break down these dead dinoflagellates, they use oxygen they extract it from the water. So as these things die off and their bodies decompose, the water gets depleted of oxygen. So the fish that were once in this water column that was rich in oxygen are now swimming around, and they're suffocating because there's no oxygen in the water column. And what happens is it leads to what's called red tides. You can see dead dinoflagellate populations in the water because of all the red color. That also tells you that the water doesn't have any oxygen in it, and as a result, you'll see just large populations of fish start to end up on the beaches because they've suffocated to death. You'll see their gills are kind of a maroonish color. Anyway, it's terrible. Dinoflagellates, photosynthetic, our example of alveolates. We'll see 2 other examples of alveolates. One is called AP Complexions, and just let the name be kind of descriptive about these organisms. They have a complex life cycle, as their name suggests. It often involves more than one host. What I want to say about the 2 separate hosts at this point is that one host has a closed circulatory system. So all the blood is enclosed in vessels. This would be mammals. We have a closed circulatory system. But the other half of the life cycle in these AP complexions involves some type of insect, typically an organism that has an open circulatory system. The mosquito is an insect. It has an open circulatory system. This is a grasshopper, not a mosquito, but I used it earlier as an example of an organism that has an open circulatory system. Those little spiracles just allow oxygen to diffuse into this fluid. There's typically higher oxygen levels in hemolymph. So where we're going with this when we get into eukaryotic life cycles, we're going to see that it's the stage of the open circulatory system where these organisms are going to basically hatch and grow into adults. And then once they grow into adults, we're going to see that they feed on blood, and their whole reproductive cycle happens inside of a closed circulatory system. So they alternate between open circulatory and closed circulatory system. We'll see several examples of that when we get into these AP complexions. So don't think that this Plasmodium is the only example, but it is our first example in which we see complex life cycles that involved open circulatory systems followed by closed circulatory systems. Okay. Our last alveolate, the one that we see in lab, these are the ciliates. And as their name suggests, they have these little tiny cilia that cover the surface of these organisms. They can use that cilia for motility. We'll see them underneath the slide moving around. There's our paramecium. That's an easy one to find. We also have fresh examples of this one that's called a stentor. And both the stentor and the paramecium have these little rows of cilia that they can use not only to move around, but they use them almost like that Roomba uses, if you've ever seen like the underside of one of those Roombas, they've got these little brushes and they're constantly just trying to pull things into their little chamber. That's kind of how this stentor works. It's anchored itself to the substrate, in this case some kind of scum in the pond water, and it's reaching out with its kind of Roomba-like mouth and those cilia to pull stuff in. So we're going to see these in pond water. I hope, obviously we've got them in jars. We're going to find paramecium and stentor in jars, and then I'm hoping to find them in the wild in the pond water that we bring in next time. So we're looking for this. And that structure right there that kind of looks like a little star, that thing is still shaky, that's what we're going to call a contractile vacuole. And if this cell moves into a hypotonic solution and it starts to swell and take on too much water, this contractile vacuole can contract and force out excess water. So it prevents it from taking on too much water that it's ever going to do damage. So those are the three examples of alveolates. Again, if I go backwards in the slides, we've got dinoflagellates, we've got AP complexions, and we've got ciliates. The dinoflagellates were the only photosynthetic ones. Okay, next one on the list is our clade of protists that we call the rhizarians, or just rhizaria. There are two rhizarians that we will see in the post-lab quiz or on the exam. The first one is called foraminiferans. I'll just show you the picture of the next one. Here's a picture of the next one. You've already seen that picture before. The second picture is called radiolarians. And I'm going to go back and forth between the two because radiolarians, you can see that what this organism actually is. It's a delicate little amoeba-like structure. It's a single-celled organism that would be very vulnerable if it weren't for the fact that it has built its little protective shell that we call a test. It has built its little test out of silica materials that it's pulled from the water column. So it's kind of built its own little glass house for protection. If I go back to the previous slide, here is that foraminiferan. It too is one of these little delicate amoeba-like structures that would be vulnerable to predation if it didn't build this little protective structure called a test. This test is made out of calcium carbonate. So more like the chalk, like if this was a chalkboard, chalk is made out of calcium carbonate, or maybe the better example of calcium carbonate is limestone rock. If you can close your eyes and picture a limestone rock, if we could look at that under high magnification, all limestone rock is the dead bodies of these foraminiferans that have been compacted under a column of water. Not that this is a geology class, but just for a second. I had some buddies over the weekend, they were saying something about they were going to Glen Rose. They were taking their kids to look at the dinosaur fossils that were in the... Has anybody ever seen anything like that or gone out to that area? No big deal. It's all right if you haven't. What's crazy is that there are some layers of limestone, like that Glen Rose limestone, that is impermeable to water. It's like this countertop right here. If I poured water on that, it's so solid that water is not going to soak into it as if it were a sponge. And limestone, like that Glen Rose limestone that's impermeable, formed under really deep seas. The water column was so deep that as these foraminiferans lived their life and they died, their little calcium carbonate test drifts to the bottom of the ocean. And if the water column is really deep, it compacts those dead foraminiferans into a solid layer of limestone. So the Glen Rose limestone formed in really deep seas where it became compressed, and it's now like a solid slab. It's impermeable to water. But ocean levels fluctuate, and if you go over to the Austin area, there's the what they call the Edwards layer of limestone, and the Edwards limestone formed also in warm, shallow seas, just like we talked about all limestone forms. But if if the seas are not very deep, if it's a shallow sea, then there's not that much weight to compress those foraminiferan bodies down, and you end up with limestone that's softer, it's porous, it's like a sponge. And we will see that water carries minerals that can dissolve little pockets in limestone rock. So it becomes like a sponge. That's where those little underground salamanders live, in those little pockets. What's also interesting is that those little holes that are in the limestone that I'm talking about, those holes are constantly opening and closing off. So you get these underground populations of salamander that become geographically isolated from one another. So you get all of that allopatric speciation that happens down there. I don't know why we're talking about limestone formation. I guess because we mentioned foraminiferans, and this is basically where limestone comes from. It's the dead bodies of foraminiferans. They were amoeba-like structures that had to build this little test to protect themselves. They feed by way of these tiny little pseudopodia that reach out. You can see those pseudopodia have grabbed on to something in the water column. So foraminiferans, they make their body out of calcium carbonate. Radiolarians make their body out of silica. So this also is kind of geologically important, because if you have seen anything like the first stone tools that were made. Like I found an arrowhead on my property the other day. Even cooler than that, I think it was last summer, I found this palm scraper. It kind of looks like a big arrowhead, but it's something that fits perfectly in your palm, and it would be used to kind of scrape meat away from the hide. What's interesting about these stone tools, the type of stone that makes these sharp edges, that stone is called chert. It's what flint is made out of, like a flint rock is chert rock. And if you want to think what that is actually made out of, it's the dead bodies of these radiolarians. So these also are in really high numbers in marine environments, and as that little amoeba dies, its silica shell, that test, drifts to the bottom of the ocean floor. And as those get compacted and formed into rock, this is what forms chert, which, if you break that apart, that's how you get your pieces of flint. Anyway, it's the dead bodies of radiolarians. So those are the two rhizarians that we need to know, foraminiferans and radiolarians. Both amoeba-like, they just build their test out of different stuff, either calcium carbonate or silica. Okay, moving to the next one, and that is this Amoeba Zoenclade. We will see individual amoebas. There is, I have not brought it out yet, but there is a tiny little container that says Amoeba Protist on it. And these don't have a lot of color. So when you look at these things in the little dropper, it's not going to look like you have anything at all. These are kind of hard to hunt for. So when we do our lab activity on Wednesday, you might want to make maybe 2 or 3 slides of this and look at all 3 of them under the scope. Hopefully you'll find an amoeba in one of those. Again, it doesn't have chloroplasts, so it doesn't immediately become obvious when you look at it. It is a heterotrophic organism like us. It has to hunt down and engulf its own food. And you can see it has these little, it's not a true arm, So it's called a pseudopodia, just like we saw the other amoeba-like structures have. And it's just trying to engulf whatever food item it's after and is going to use its digestive enzymes to dissolve that. So just like us, it's an ingestive heterotroph. That's a single-celled amoeba. Again, we'll see it on Wednesday. But in addition to that, what's called an Amoeba Protist, there are these larger examples of amoebas. This is a slime mold, and we do have an example of that in lab. We'll see it looks just like these yellow veins, but if you look at this under the microscope, you're going to see that those veins are just a river of cytoplasm, and it's constantly flowing, and it will keep flowing in the direction of nutrients. And if there, again, if it starts to wander in a direction where there isn't any nutrients, then it will dry up, and cytoplasm will instead flow in the direction wherever there are the most nutrients. So it grows in a structure that looks a lot like fungus. So it was originally mislabeled as a fungus. What else? There are, I think, just two more clades. We have red algae and green algae. And if you're keeping notes so far on multicellular protist, I believe brown algae was our first multicellular protist, and now red algae is our second multicellular protist. You can see this. Clearly those cells are starting to differentiate into, some are functioning like leaves, others are functioning like a stem to hold those leaves up. And we can't see it on the picture, but still other cells are going to be functioning as an anchoring structure. There, and obviously we see multiple cells there. So red algae, we'll have slides of this on Wednesday. Most of these are marine, and they live a little deeper in the water column because those longer wavelengths of light are able to penetrate deeper down in that water column. So no matter what wavelength of light it is using, it's still using it in the same way for that first step of photosynthesis, splitting those water molecules, creating this imbalance of charged particles on one side of the membrane. They have to flow across to the other side of the membrane from an area of high to low concentration. That spins a turbine and makes ATP. So red algae are doing this. Green algae are doing the same thing. They're just capturing a different wavelength of light. The chlorophytes are the green algae, and again, all of these are aquatic. They have no adaptations to life on land. So let me make some distinctions between these, because we can see in terms of green algae, some of them are single-celled, the unicellular protist. We can see some that look like this that are colonial protists. There's clearly more than one cell, but if we look closely, each of those cells are identical. They're doing the same thing. And then up at top, the sea lettuce there is multicellular. Some of those cells are anchoring to the substrate. Other cells are broad and blade-like that are doing the photosynthesis. So in lab on Wednesday, we'll see our sea lettuce, we'll see Volvox, and we'll see Chlamydomonans, this unicellular green algae. Just to connect some dots, our last lab activity that we had, we were using microscopes and we were looking at prokaryotic cells. So even under high magnification, we were just looking for differences between the shape of the cell. Is it sphere-shaped? Is it rod-shaped? Is it spiral-shaped? And now when we look inside these protists, we should see nuclei. We should see organelles. Here, just to prove to you, I got 2 slides left, but again, Volvox, we said, was colonial because we've got cells doing the same thing. And if we saw this under the scope last time, there are these little reproductive colonies that are on the inside of the sphere. And when the parent sphere finally ruptures, we can see these little reproductive spheres emerge. And each cell kind of goes to work pulling in its own nutrients and kind of lives independently. So our good example of a colonial green algae, there's our multicellular green algae, and these Chlamydomonans, just our last slide. I just wanted to show, this is going to be a segue between green algae and eukaryotic organisms. I'll grab my pointer. I've said this a couple of times. Green algae are aquatic. They have no adaptations to life on land. Plants, on the other hand, are going to start to have adaptations. They're going to start to differentiate themselves from green algae because they're going to spend more time in this diploid part of the life cycle. So here's where we kind of remember some stuff from 1406. There are parts of the life cycle. In fact, we have parts of our life cycle in which involve haploid cells, the gametes, sperm and the egg. And then those haploid cells fuse and we get our first diploid cell. In fact, we live our entire life. We are now comprised almost entirely of diploid cells. Only the few cells in our reproductive structures that are making those haploid cells. So we are comprised mostly of diploid cells. Protist, if we go back to the green algae that we're talking about here, this unicellular green algae of Chlamydomonads, this structure is haploid. And it's actually quite happy staying in the haploid form as long as there's plenty of moisture and plenty of nutrients. This thing is in a nice pond that has, again, fertilizer running into it. The pond's not drying up. It's quite happy just staying in this asexual reproduction, which is no different than mitosis. It's just making clones of itself. So this is easy to do. The problem, like we said, is it generates little to no variation. So that's problematic. We're going to see that in addition to this asexual reproduction, there is another pathway. If we go this direction, there is, this picture shows two complementary unicellular protists getting together. We can't really use the terms male or female. So you can see one of them has a negative sign and the other one has a positive sign, just meaning that they are not part of the same lineage of cells. They're both green algae, right? But they're not from the same strand of cells, basically. So they are complementary to one another. They can get together, share some of their DNA, and when they go through this sexual reproduction, they typically do this when the pond is drying up. And as the pond is dry, they kind of wait out those tough conditions in this single-celled, let me try that again. It's a single diploid cell is what I'm trying to say. What we call the very first diploid cell, that first diploid cell is called a zygote. And in green algae, they don't stay in this diploid stage long at all. As soon as this diploid cell forms, it immediately goes into meiosis and splits back into haploid cells. Each of those unicellular green algae are haploid cells, and they stay in that haploid form until the pond starts to dry up. And they need to wait out these tough conditions. Then complementary unicellular organisms to get together, they produce this diploid zygote that you can see has a hard exterior shell to it that allows it to kind of sit in these dried up ponds and wait out these harsh conditions. And then when the next rainstorm comes and conditions are just right, that zygote immediately goes through meiosis back into this haploid form. What I'm trying to say here is that there is no multicellular diploid phase. It's just a single diploid cell that doesn't last very long. Obviously, what we can see here is that diploid cell is better at surviving harsh conditions. So when the pond dries up, that's when they switch over to this diploid cell, and they really just wait for conditions to improve. Plants are not going to wait for conditions to improve. Plants are going to start to prolong. They're going to extend this diploid phase of their life cycle. May be obvious, but it's just it's an interesting transition from how plants, they evolved from green algae who at one point spent all of their life cycle in the haploid phase. And as plants adapt to life on land, they move away from the haploid phase and spend very little time in it and end up spending most of their time in the diploid phase. Okay, so that's it in terms of new lecture. We will see eukaryotic life cycles in a little bit more detail on Wednesday. We're also going to do our Protist Diversity lab activity where we're going to get our hands on most of those organisms we saw in our short lecture.